I'm going to talk about controls of microRNAs and siRNAs during embryonic development. And I want to start off by thanking Marv for introducing the subject of the importance of motherhood, and on behalf of my mother, as well as the behalf of my talk. And also to thank Donal for introducing some of the ideas about microRNAs and siRNAs. And so I'll be building on those. This is work done in my lab with the aid of Elizabeth Lund and Mike Sheets, who's another professor in my lab, or in my department, a former graduate student of, with Marv Wickens, actually, and technician Suzanne Imboden. So the experimental system that we work on is Xenopus laevis. We're talking about very early development in the Xenopus lifetime. You've already heard from Marv about Xenopus, and there's the entire time of my talk is just going to be preoccupied with very early events down here, just before and after fertilization. Okay, as you heard, the oocyte is a very important cell that develops, that puts various messengers and so forth into the egg in the right way, and then after fertilization, there's a period, an extended period of, where there's no transcription to speak of. DNA is replicating, but it's not being transcribed. Very active cell division, and then out here at about, depending on the temperature, maybe 8-10 hours, there's the mid-blastular transition where transcription starts in a large level. The transcription that starts at that point is the synthesis of zygotic genes, the embryonic genes. And as Marv told you a few minutes ago, up to that point, there's a lot of protein synthesis going on, but that protein synthesis is encoded by messenger RNAs that were stockpiled in the oocyte during oogenesis and matured into active forms in the egg. So the assays that I'll be doing, a lot of gel assays, looking at RNAs isolated from different stages of these embryos. One of the reasons that Xenopus is such a nice system to work in is that the oocytes are actually fairly large, and you can do microinjections either into the cytoplasm or into the nucleus, and use it as a little test tube. And that's what we do a lot, is we inject either DNA or RNA into various components of compartments of these cells and watch to see what happens. I said there's very little transcription until the mid-blastular transition, when zygotic transcription turns on in a large way. Prior to that, actually in about 3 or 4 hours after fertilization, we do see some transcripts being made. And when we analyze those transcripts, they're primarily a very large molecule that subsequently gets processed into a precursor of microRNA, and that precursor then is matured into the mature microRNA, and for the first several generations of the embryo, this is the predominant microRNA in the whole organism, and this is called miR427. There's a similar system that has been described by Giraldez and his co-workers at Yale. That's in zebrafish, and that's, I think, miR430. It just happens to be numbers that are similar to each other, but the consequences are fairly similar to what we observe in the frog. So this is one thing that happens. We do see the formation of miR427, but we also see other things happening in RNA metabolism. And not only do messenger RNAs get polyadenylated and elongated by the addition of poly-A, but some of the messenger RNAs, a specific subset at the mid-blastular transition, start to lose their poly-A. It's not that all messengers do, but it's just a subset, and these are maternally encoded messengers. In this case, cyclin B2 messenger RNA. There are several others, but this is the one we have focused on as a typical. So you can you see this is the non-polyadenylated RNA, and with time it gets bigger and then shortens after fertilization or after mid-blastular transition. So we asked whether the accumulation of the miR427 might be associated with this reduction in the poly-A, and the reason we did that is it's known from our work and others that microRNAs can lead to deadenylation of messenger RNAs. If there's a target region in the coding in the 3' UTR, microRNA will base pair with the target, carry on with Ago proteins in, which will recruit deadenylation machinery and translation inhibitors, and you'll either get translation repression or degradation. So what I'm going to be talking about is evidence that miR427 participates in this event, but I'm going to also talk about other events that go on early in development with regard to siRNAs and the proteins that are associated with the microRNAs. I was standing next to Glauco during that 20 minutes or something. We were waiting for the photograph, waiting for Dino to come, and all of his cigar smoke was coming right through here.
Wrong diagnosis.
So what we do is we use a model system instead of using the cyclin, the entire cyclin messenger RNA. We've taken the 3' UTR of this messenger RNA and put it downstream of a beta globin gene. It's just an easier assay for us, but what we're interested in, as Marv was emphasizing, we're interested in the sequences that are in the 3' UTR. And there had been an element early on, defined early on by Rebecca Hartley and Mike Sheets, saying that somewhere in this area there's a deadenylation element, and so we looked to see whether there was a sequence that could be complementary to miR427, and you can see that's shown right here. And as Donald told you the other day, the interaction between microRNAs and their targets is not perfect. In fact, almost always there are bulges in the so-called duplex. So here's the seed sequence that is absolutely critical for that kind of interaction. But there are oftentimes we'll see other kinds of interactions, and we can see something there as well. When we just look at this, we inject this transcript into the oocyte or into the 1-cell or 2-cell embryo, and then watch to see what happens to it. And this is what we do. We put in a non-polyadenylated RNA. This happens, what we put in has the signals that Marv told you about in his lecture this afternoon, the CPE for cytoplasmic polyadenylation and the A2UA3 for the poly-A. And you can see that the transcript that goes in gets bigger, and this is because of the addition of the poly-A. And then with time, it decreases in size, okay, as the poly-A is removed, just as it is with the endogenous RNAs. So we asked whether this sequence here on the in the circle is required. And so in that case, what we've done is we mutated the complement of the seed sequence, so it would no longer be complementary to the seed sequence. And now the RNA is stable, as you can see here. So we conclude that, in fact, this interaction is essential for the deadenylation. And so it says that this entire 3' UTR has only one site for miR427, but that one site is sufficient for the deadenylation. You've already seen this kind of schematic. In the case of miR427, the primary transcript is very, very long. We see it as just a very long smear on a gel, but it contains repeated hairpins, and these hairpins each contain a duplex, one strand of which is 427. This is called pre-miR427. It's exported from the nucleus of the cytoplasm by exportin 5 in a RAN GTP-dependent manner. In the cytoplasm, one of the strands is removed as it binds to dicer, and the top of the microRNA is, the precursor is removed, and we end up with a mature RNA. Now what that does is, when we look at the poly-A itself in the, on our test RNA, we see that the poly-A shrinks with time. Now, what we've done in this case, in this experiment, is we didn't just wait for the cell to make the miR427 during the early development. We injected into the early embryo, we injected the precursor itself to see whether that would accelerate the deadenylation. And that's what you see here. We accelerate the deadenylation by several hours compared to, this is without any exogenous precursor injected, and here is with the precursor, and you can see it's several hours faster. So we're able to accelerate by putting in the precursor, and this is just the assay that we used. And so it says that at least to some extent, the deadenylation is limited by the availability of the microRNA. So what that is telling us is that this is a kind of a switch that is turned on very early in development. Once this particular messenger RNA is no longer required for for the development of the embryo, the cell wants to get rid of that particular maternal message and go on and make other messengers that will be important, of course, in the normal development. We also looked at the expression of Ago proteins. You've heard about these. These are the proteins that bind to the microRNAs. And over here on the left, you can see if we just take oocytes and look at Ago proteins, this is by a northern blot, I'm sorry, by a western blot, looking at the Ago proteins, you can see that there's quite a bit of Ago protein in the oocyte, okay? But this, when one isolates oocytes in this system, as there are cells, which Marv referred to before as nurse cells that feed the developing oocyte. And these nurse cells themselves are actually mature cells. And so we had to, if we strip off those follicle cells, the nurse cells, then we find that almost all of the Ago protein disappears. So even though it appeared to have a lot of Ago protein, those were not in the oocytes themselves. Okay, so it's just a little bit of a cautionary tale that it's a good idea to question whether what you're working with is what you think you are working with. When the oocyte matures, then it does make its own Ago protein, and then we can see an increase here. Okay. Over on the right-hand side, we see that not only does do we see an increase in Ago proteins during maturation, but when the egg is fertilized and starts developing, we see the appearance of quite a bit of more Ago proteins, especially after the MBT period. Okay. There's also an increase in the messenger RNA for Ago2. We don't see a large increase in messenger for another Ago protein, Ago4. So there seems to be developmental control in this system. Now, that's all I want to say about the microRNA 427. It comes on, and the Ago proteins that are needed for its function also come on very early and are involved in the deadenylation of targeted maternal messenger RNAs. Now, I want to change my subject a bit here and go from microRNAs to siRNAs, the small interfering RNAs. Okay, and again, as you heard earlier, siRNAs normally target their or bind to their target by forming a perfect or almost perfect match with the targeted sequence. And often that targeted sequence is in the coding region of a messenger. Okay, it doesn't have to be, but we often see it there. And in other systems, it's been shown that Ago2, one of the 4 Argonaut proteins that Xenopus has, Ago2 has an endonuclease activity, and that Ago2 slicer activity that you've heard about is responsible for cleaving the target RNA. And since the siRNAs are about 22 nucleotides long, miR427 is the same length, and they all seem to bind these Ago proteins. We asked whether the endogenous miR427 could act as a guide in siRNA cleavage of a target. And much to our amazement, it does not. This is the experiment that we performed on that. We generated, again, a model substrate and model target, got rid of the target that would otherwise bring about the deadenylation, because we didn't want that to complicate the experiment, but made a perfect match for 427 in the coding region here. And then we asked whether, if we inject this RNA into embryos, does this RNA get cleaved? And as you can see, it's perfectly stable. So this target does not serve as a cleavage target for siRNA. So we asked, well, maybe there's something funny about the particular endogenous miR427. So we put in a double-stranded miR427 that would act like a precursor of siRNA, and asked whether that would also work, could work, and that's shown here. And again, we got no cleavage of the target up here. So then the next possible explanation is that maybe we're missing an Ago2 activity in these embryos. And so what we did was to take a clone of human Ago2, made messenger RNA from that human clone in vitro, injected that into the embryo, and when we did that, we call this Ago2, inject that into the embryo. Now we see cleavage of this RNA here, and these are the 2 fragments that are generated. So what that says is that the embryo lacks Ago2 activity and is unable to carry out siRNA cleavage. This cleavage occurs only in the with the wild type Ago2. If we inject the messenger RNA that encodes an Ago2 protein that is defective in its cleavage domain, then we see no cleavage here. Okay, so the embryos, as far as we know, probably has Ago2 protein, but it does not, because at least we can see messenger RNA for Ago2 being made. But if it's being translated, into the protein is inactive. And we don't yet know the reason for that. So what this says is that at least we can carry out siRNA targeting of specific messenger RNAs in this system, but to do so we have to supplement it with exogenous Ago2 messenger or protein. I mentioned that as far as we know, these embryos do have the Ago2 protein, and another test that we carried out to see whether there was any activity, cleavage activity, is related to something that Dahlberg talked to you about at the beginning of this course, and that is he and his lab have worked on a microRNA that's processed and matured by not by the usual Drasha-Dicer kind of processing that I explained a couple of slides back, but Ago2 actually is able to process one specific precursor RNA that of a microRNA called miR451, where the microRNA is all on one side of a hairpin and it goes around the loop, and the Ago2 cleaves in this case. So this is a direct assay for the Ago2 cleavage activity. And when we carry this, made this substrate, put it into the embryos, it was not cleaved, it was not processed, it stayed as the full-length molecule. However, if we supplemented the embryos with the human Ago2 that has cleavage activity, now we do see the cleavage of the sort that Dahlberg talked about the other day. Okay. So again, this says that the protein is, well, we think it's there, but it does not have any activity. And this is just supplementing the control, supplementing a mutant human Ago2. We did the same sort of experiment in oocytes, and not surprisingly, we see no evidence for for cleavage activity in the oocytes. It's not surprising since we didn't see it in the embryos. So what we've now done is we say, well, we can use our system now to target messenger RNAs that are present in the early embryo. And this is something that hasn't been possible in the past. People have tried to do RNAi targeting of messengers and without success. And excuse me. First control we wanted to do was to see whether just putting Ago2 into an embryo affects the embryo, and it appears they appear to be perfectly normal. Okay, so that's good that we can make these guys happy. So now we can do our real experiment of asking about siRNA. And this is a few embryos that resulted from putting siRNAs into them directed against particular messenger RNAs that are involved in early development. And you don't have to be a developmental biologist to see that these things here and here are very different from the normal embryo. So what we are seeing is that there is specific disruption of the developmental pattern, and in ways that seem to mimic some of the phenotypes that have been seen using other methods, knocking out, say, this particular FOXD3 or the vent genes. So this gives us some assurance that we are able to break down, to inhibit development in the system. However, there is another problem here, and this is something that we need to be a little careful about, and that is that siRNA early on can affect later development. So there seems to be an effect, an indirect effect, or at least an effect that we didn't anticipate from injecting the siRNA. So it doesn't show up terribly well, but these guys, these embryos, tadpoles, are smaller, the brain is smaller, eyes are smaller, and so there seems to be neurological problems here. And this is just injecting a non-specific double-stranded RNA into the embryos. So what stage or what effect is, or what are we affecting with this non-specific double-stranded RNA? First thing we wanted to do was ask whether the normal process of making microRNAs might be affected, since we're putting in a fair amount of double-stranded RNA. And this is the experiment to test that. This is the 427 that you saw before. And in normal cells or normal embryos, it gets processed into the mature miR427. However, if we inject into those embryos a non-specific siRNA, you see that all of the synthesized pre-miR427 stays as the precursor, and it does not get processed into the mature RNA. So that means that the double-stranded RNA in this system is inhibiting dicer and dicer activity, which is sort of the last cleavage step in making the microRNAs. Okay. 427, as I told you at the beginning of this talk, is made very early in embryogenesis. In fact, the transcript that synthesizes it is made before the mid-blastular transition. There are other microRNAs that are made in later times in embryogenesis, and when we look at those, for example, miR19, 19B, and 16, we see that in those cases the maturation of these RNAs seems to be pretty much normal, even in the presence of the double-stranded RNA. And so that says that there is something made at about somewhere in this period in the development that suppresses or overcomes the inhibition by the siRNA. Okay? And a good obvious candidate might be Ago proteins. And so we asked whether we could overcome this inhibition by generating different Ago proteins in the embryos. And the answer is yes, we do. This is the control where there's no siRNA. Lanes 3 and 4 is when we put in the siRNA, and you see that maturation of the mature miR427 is inhibited. And then by putting in messenger RNAs for 3 different Ago proteins, now we get normal maturation of the microRNAs, of this microRNA, even in the presence of the inhibitor. So that shows that the that Ago proteins are somehow maybe titrating out, binding to the excess siRNA, double-stranded RNA. It's not just a double-stranded RNA binding protein that's doing this, because if we put in TRBP, which is also a double-stranded RNA binding protein, but works totally differently from any dicer activity, that does not rescue from the inhibition. As expected, this is also sensitive to the deadenylation effect is also sensitive to the siRNA inhibition, but we can rescue that again by putting in Ago protein. In this case, the mutant form of the Ago protein, Ago2, which does not have the cleavage activity, nevertheless rescues the production of the miR427, which says you don't need cleavage; it's just binding to the siRNA. This is just another control. So multiple Ago proteins can rescue dicer inactivity and give us deadenylation, even in the presence of siRNA. So what I've shown you is that the accumulation of miR427 and Ago proteins is developmentally controlled. Both oocytes and embryos require the expression of Ago2 in order to carry out RNAi. So experimentally, if you want to do RNAi in this system, you need to put those things in. Mainly it's the Ago2 and the siRNA that you want to use. Just as parenthetically, I mentioned that zebrafish have a comparable system in deadenylation by a microRNA that's made very early after fertilization. And in zebrafish, again, they cannot carry out siRNA-mediated targeting of messenger RNAs. So it would be interesting to try this, see whether putting the Ago2 messenger RNA into zebrafish embryos would allow that to happen, to occur. Because, I mean, zebrafish especially is a very powerful experimental system that a number of labs have used, but there is the difficulty of not having the siRNA system up and going. And finally, siRNA just non-specifically can inhibit dicer, so one has to be aware of that. Where we're going, questions to be asked is asking about the regulation of Ago2. Why don't the cells have it? We don't know. What step in the dicer's activity is inhibited by siRNA? And as I mentioned, whether the zebrafish embryos can also benefit from this kind of technology. So with that, I'll stop and take questions. Thank you.